Group 5. We're going to be leading this week's discussion. The title of the paper is Genital Interactions During Simulated Copulation Among Marine Mammals. My name is Nathalie Alamar, and I'm a fourth-year zoology student.
Hi, I'm Noah Millicent. I'm a fourth-year marine science student.
I'm Natasha Salas, and I'm a fourth-year marine science student.
And I'm Caitlin Torres, and I'm also a fourth year, majoring in animal biology and minoring in zoology. So why did we choose this paper? It basically gives us a better understanding of how sexual selection influences the shape of genitalia. And this paper was pretty relevant to our assigned chapter, which was chapter 5. And this paper was interesting to all of us because it allowed us, and hopefully all of you, to understand how marine animals choose their mates and why they fit. So here's a little bit about the authors. 4 different writers contributed to this scientific article. So the first one is going to be Dana Orbach, and she's an assistant professor of marine biology at Texas A&M University and is a comparative biologist. And then Diane Kelly is a biologist that splits her time between studying sex and writing about science. Her research focuses on reproductive anatomy and function. And Mauricio Solano is a veterinarian, radiologist, and teacher at Tufts Cummings School of Veterinary Medicine and has a passion for scanning marine animals. And Patricia L. Brannon graduated with her PhD from Cornell University and has a specialization in morphological evolution of reproductive structures, sexual conflict, and post-copulatory sexual selection.
Okay, so now I'm going to go over a little of the background to the study to provide you all with some context. So as mentioned last week, there's a lot of diversity in male genitalia and a lot of studies being done. However, female genitalia has been kind of neglected and sparsely studied. Despite this, we are seeing that there's quite a bit of diversity amongst them. Now, this paper highlights the fact that there is little known about which features of male and female genitalia interact and how do those interactions affect fertility. As they build up to how they will answer this, they mention the different techniques that have been used across species to demonstrate a close fit between genitalia and mechanical interaction. Moving on to our studied creatures, some of which you can see here on the slide, marine mammals have unique adaptations influenced by sexual selection and environmental constraints. Specifically, the cetacean vagina contains muscular protrusions of the vaginal wall that can vary, and this includes in number, shape, size, and positioning across species. And this morphological diversity may be driven by sexual selection. Another area that the authors were interested in is whether the terminal tip also penetrates through the cervix. Now, the authors had 4 main objectives: to determine shape compatibility of marine mammal genitalia. The second was to examine potential navigate through the complex folds, spirals, and deposit sperm deep in the female reproductive tract. The third is to infer the copulatory position that would result in the best genital fit between the sexes. And the last is to develop a technique to assess copulatory fit in large vertebrates.
To begin, so the methods that they used for this study began with specimen collection. They did this by finding intact reproductive tracts taken from naturally deceased marine mammals through a network along the coast. They needed to be fresh, less than 24 hours post-mortem, and sexually mature. Once they were found, they were immediately frozen and then sent to the lab. This included male and female harbor porpoises, common bottlenose dolphins, short-beaked common dolphins, and harbor seals. Firstly, with males, all male reproductive tracts were cleaned, photographed, measured, and weighed as they were. Then from there, they were pumped with fluid to stimulate an erection. At this point, they were then remeasured and then submersed in a solution to fix them in this distended state. As for the vaginas with females, molds were created of the female reproductive tracts, and from there, the penises that were distended in their fixed form were inserted into the vaginas. The team then was able to determine the best shape compatibility and sew them together in that place. From there, 153 photos were taken of each specimen pair in order to produce 3D models for them to use further in the study. Because of the differences in shape and volumes in relation to the vagina, different alignments and other things with different species. It is not something that can be cross-referenced or generalized between different species.
So ultimately, what the research team discovered was that genitalia in these male and female mammals closely evolved together. There were two types of coevolution discovered, and these were congruent and antagonistic coevolution. Congruent coevolution was found in short-beaked common dolphins and harbor seals, where there were no physical barriers within the vagina, so penetration was not inhibited. Antagonistic coevolution, on the other hand, included physical barriers in the form of vaginal folds that inhibited penetration, and this was found in common bottlenose dolphins and harbor porpoises. The largest vaginal fold actually bends the penis backwards to prevent fertilization. In this way, vaginal folds are thought to be a mechanism of post-copulatory control of paternity. However, the male marine mammals have a way to bypass this system, as it is thought that due to voluntary control of the penis and live cetaceans, it can avoid the vaginal folds. Even so, there was no evidence that the penis penetrated the cervix, so the sperm is still subject to chemical, microbial, and immune response defenses. There's a diagram on the bottom. On the left is the harbor porpoise. On the right is the harbor seal. These are both CT scan images of the penis inside the vaginas of the mammals. The red arrow points to the vaginal fold that prevents penetration in antagonistic fits, and the red areas on both sides. On the right side of both the images are the presence of erectile tissue. So body position is a very important part of successful copulation in marine mammals, especially for antagonistic fit species. The penis has to be pointed upwards to deposit the sperm near to the egg, so the optimal position is a dorsal penis to ventral vagina orientation. For a congruent fit, the closeness of the male urethral opening to the opening of the female cervix is best. The closeness is best, which is at the angle of 0 degrees. The importance of this study is this method of inflating intermittent organs with pressurized saline could be used to learn more about the genital morphology of other species. These techniques could also be used to explore copulatory fit and predict which body positions and copulations will lead to successful fertilization. And finally, it can be used to aid in captive breeding programs for species conservation, mainly for sperm collection and stimulating fertilization in females.
So now that you've heard a bit about our study, here are some of our discussion questions. Though the best quality of male and female reproductive tracts were chosen as models for each species, was the sample size too small to justify the results from this experiment? In what ways is the morphological structures of the genitals driven by sexual selection or the environment? Is there any specific marine species that you would like to see this methodology done with, and why? And can you relate this to our current chapter or any of the previous ones? We hope you enjoyed our journal.
Thank you.
Thanks.